Okay. Hi everyone, how's it going? My name is Nathan Lord and I am an entomologist at the LSU AgCenter, the Department of Entomology at Louisiana State University. And today we are on location at the exhibition, an art and science exhibition that my lab and I and several other collaborators that we'll talk about have put together. And it's titled Iridescence. So when you walk up the stairs, this is the first piece that you see, and this is a photograph created by one of my collaborators and dear friends, Jennifer Robison. And this is life-size, so this is actually her daughter, Ella, who took one of my entomology classes at LSU. And Jennifer is an instructor of photography at Bossier Parish Community College up in Shreveport. And she had an interest in entomology, specifically these beautiful, radiant morpho butterflies. And it was through Ella's entomology class that we met, and I got to see some of the amazing works she had done. And so Jennifer and I started talking about how we might be able to bring iridescence and the beauty and the science and the art of iridescence to the public. And so this work here is, there are 97 real morpho butterflies that she spread individually. The genus Morpho is found throughout Central and South America, and some of the most well-known examples of iridescence. And so they have these butterfly scales all over their wings that reflect light through constructive interference, and then you get these extremely vivid colors. But what's interesting about iridescence is that it changes as you move. And so all of the works in this exhibition, depending on where you are standing, depending on how the light is coming in, they will all change in and of themselves. And so one of the larger themes of this exhibition is perception, and how, although we study the evolution of color and vision of insects in our lab, that's based strongly on the perception of the organism viewing it. And so as we're most familiar with the human visual system and maybe some anomalies of the human visual system like color blindness, it's sometimes surprising for people to learn that all sorts of other organisms have visual systems that extend far beyond what we can see. And so if you think about flowers, actually have UV patterns that we can't see, that only certain insects can see, that bumblebees land on. Birds can see in the ultraviolet, and a lot of insects can even see out into the near infrared also. So a lot of the works here are both of the natural variety, and a lot of them are also of the man-made variety. So how this exhibit came to be was I had a grant from the National Science Foundation on studying the evolution of color and vision in this one group of beetles called the jewel beetles.
And can you introduce yourself for new people?
Sure, yeah. So my name is Nathan Lord, and I'm an assistant professor at LSU in the Department of Entomology. So it occurred to me, because having worked on insects for most of my life now, most insects are boring, they're brown, they're tiny, they don't look spectacular. And it just so happened that the group of beetles I was working on tended to draw the attention of everyone. And I thought that if that was the case, then I should probably use that opportunity to be able to educate a little bit about the science that we do. And so we had an idea to actually create an art and science exhibition that explored both the science behind the principle of iridescence, but also showcase the art. And so I was able to strike up a collaboration with the Louisiana Art Science Museum downtown Baton Rouge. And much to my surprise, they were super interested in the idea of a bug doctor creating an art show that, I had no business playing in that sandbox. But they thought that the idea was wonderful. And a year and a half later, here we are. And we've got pretty much the majority of an upper gallery in the Art and Science Museum right on the Mississippi River. It's got an invited portion where we invited artists from all over the world to submit their works. It's got a juried portion, which means that we had an open call for works that featured Iridescence, and a juror that was selected by the staff here got to critique and pick the entrance for the juried portion, which is on the other side there. And then we have a room specific to our work at LSU. So that's showcasing largely the work of my awesome graduate students and what they're doing for their research to understand the purpose and the function of iridescence.
Cool. Can you show us around?
Yeah. So then you walk upstairs and you have the title wall, which we have a printing company here in town that does a lot of the graphics for LSU's, Vivid Printing, and they were able to do this wonderful iridescent work. And so this is just the title wall that features all of our awesome sponsors and collaborators and partners like the AgCenter, National Endowment for the Arts, Olin, Entergy. The Ebb and Flow Festival was able to get one of our featured artists here for our opening night, and of course we've been able to generate support through lots of other avenues as well. So here's a title wall that just tells a little bit about the works that are here. So now we'll just give a quick tour around and talk about some of the works. So these pieces are two more pieces by Jennifer Robinson, and she likes to incorporate actual insects into her works. And so she actually asked me to make some wood frames that had some...
Okay, we're back.
Yeah, so she asked me to make some wood frames for her that showcased some special meaning to these works. And so one of my COVID hobbies was getting into woodworking. And so this wood is ash, and it's native to Louisiana. And what you can see in all of the edges of this are these very intricate tunnels. And these are actually galleries of wood-boring and wood-eating insects. And so we tried to keep the frames rustic. And the reason I picked it was because that's how I got started on all this research in the first place, was researching the emerald ash borer, which is a jewel beetle that has been decimating North America's ash trees. And so that's why I started any of these projects, is to start to better understand how these jewel beetles see and why these bright green colors might be used as a visual signal for the beetles.
Are those jewel beetles on there?
So these are a different species of jewel beetles, but they are, yes. So this is a work where Jennifer took hundreds of individual beetle wings and glued them to her daughter's back. And so it gives this very almost reptilian, scaled dragon-like look. And so then she wanted to spread actual beetles and have them placed over the work itself. So yeah, really cool. And then if we go to this side, we have works by Carl Gaff, and Carl is a professional artist who is also a scientist as well, and his work deals with the property of birefringence. And so these are actually, they're not paintings, they are images themselves, and he works in crystallography. So he is very interested in how crystals form from various materials, both the structural properties and the organizational properties. And so all of these are various elements that are crystals, and these are actually images that have been shot through special light. And so while we think of light having lots of different wavelengths, lots of different colors, light also has other properties like polarization. And a lot of the sensors and cameras that we use can actually pick up on some of these properties. So similar to how you might put on polarized sunglasses and see different patterns on various windshields that have protective films, or on plastics or CD cases, that's sort of what you're experiencing here. So these works look in a way very phantasmical, but they are actually just light that is perceived through elements of polarization, and as these wavelengths of light interact with one another.
Jolie says jewel beetles were used in Victorian dressmaking.
That's right, and we have one of those dresses that we'll show you in just a few minutes. So this is one of the most incredible pieces, and this was the last install that we had for this exhibit, and this is a sculptural work by an artist named Sue Sunny Park, who uses materials to showcase the difference in perception. And so she likes to use elements like chain link fences and glass, and this particular work is obviously a very 3D element, and the beautiful part is that all these pieces of glass are at different angles, and it's called, again, it's another birefringent or dichroic material, to where if you look at a single piece, as you move and as the light changes angles striking that piece, as the reflection of the light changes the angle that it takes to get to your eye, the color changes. And so each of these pieces of glass, depending on where you are, can exhibit properties of the entire rainbow. And so then, when you combine that with this 3D element, you can see the reflections all over the place, and this will look entirely different depending on the viewer, depending on where you're situated, depending on the light. And so this is one of Sue Sunny's works that is meant to be an immersive experience that shows how perception really can change depending on the viewer. So then over here, we have works by Chris Marley, and Chris Marley is really well known in the entomological circles because he used to start out with primarily insects, and now usually most of us entomologists have a Chris Marley calendar on our walls because he does these fantastic symmetrical prism-like displays of live specimens. And so he graciously contributed these works from his biophilia exhibit. And again, the meaning of biophilia is nature-loving or life-loving. And what he tries to showcase in his works that now extend far beyond insects, but also get into things like shells and minerals and even vertebrates like birds and snakes. He sources these things either himself on field work, or he contacts collaborators that are obviously naturally sourced, and he builds these incredible works that showcase the beauty of nature around us. And so whether it's morpho butterflies, or in this prism here, this is a display of different electric blue butterflies. This is called cerulean butterflies. But whether it's jewel beetles here, most of these here are jewel beetles, the family that we study, with a few other exceptions. Or whether it's damselfly wings, as in these, he really has an incredible eye for curation. I mean, in our research group, we've spread thousands and thousands of insects, but the way that he makes them symmetrically perfect and the way he perceives the layout is to me, one of the exceptional parts.
Would you mind telling Colette asked if this is being recorded? Can you tell her it's going to be found on the YouTube channel?
Sure.
Later after this?
Yeah, so this recording will also be found on the YouTube channel after this if you want to view it later. So here we have examples of more iridescence in the natural world, and here you've got hematite. So hematite can be found throughout the world, and they have this thin layer of nanocrystals on the outside that actually give it that iridescent appearance. And so here is just, again, a couple of his amazing works that showcase the natural beauty. And again, none of these are painted, none of these are altered in any way, there's no special lighting. These are the true colors of these organisms. And so the next question that you usually get is, well, why? Why would they be colored in this way? And the simplest answer right now is we don't really know, but that's why we're doing our research. You know, again, keeping in mind how diverse insects are, how beautiful their coloration can be. You know, it's our goal to figure out, who are these colors for? Are they for the insects themselves? Do they act as some form of camouflage? Do they act as some form of signal to ward off predators? Lots of work has been done on the birds and some butterflies, but not a lot in the insects. And so this is an example here of an African starling that has these beautiful blue-green iridescent feathers. And then you get one more example of shells. You find lots of the nacre of shells, abalone and things like that. And this is abalone here, where you can see these natural patterns that are formed through, just like a pearl, where you get various thin layers of excretion from the shell itself. And those thin layers stacking on top of each other like a sandwich allows the light to interact and create these rainbow patterns. So these next series of 4 works are really, really cool. They are by an artist named Francisca Schenk, who's in the UK, and she is a painter and an instructor of art. And so I actually met Francesca when I was at a scientific conference in Cambridge a handful of years ago, and she was interested in the physics of iridescence and color. And what she found was that none of the paints that were as close as she could find to approximating iridescence were good enough. And so she turned to nature to try to figure out if she could create something that is like paint but not, in the sense that the paint actually displays the correct properties of how iridescence would be in nature according to laws of optical physics. And so this is just a series of 4 to where you can see that it goes from a sketch of this particular butterfly to applying the first layer of iridescent paint to the finished picture. And when you really zoom in, just the quality of the sketch, especially from when you then take a step back, it looks like a photograph. And truly, as you move from left to right, you can see how that purple iridescence comes in and out of focus.
You're saying that's beautiful.
Yeah, it's incredible. These are just super stunning, and it's not something you recognize until you're at just the right angle, and all of a sudden this typical butterfly becomes electric purple, and it's actually pretty startling. So on this wall here, we also have a video provided by Ten Kidsman, who is he is a scientist and photographer at the Rochester Institute of Technology, RIT, and what this is showing a thin film. So iridescence can be formed through a couple of different ways, and one of those ways is by thin film reflection. And so just like a soap bubble or an oil slick on a wet day on black asphalt, what you see is you get layers of chemical, in this case a soap bubble, that vary in thickness ever so slightly. And light enters and bounces off of what's normally a clear surface, and sometimes goes through, sometimes it goes through and bounces back, sometimes it just bounces off initially. And when you get lots of photons of light doing that at the same time, you get what's called constructive interference. And so while a particular particle of light may be red or green or blue in terms of its wavelength, when you have all of those particles of light interacting at the same time with one another, that's how you get this constant rainbow shimmer. And so that's what you see in bubbles, and this is an example of thin film. So if we walk back here, we have some very cool works by an artist and scientist named Kate Nichols. So Kate Nichols is a TEDx fellow. You can watch lots of her fantastic videos explaining the science and her art on YouTube. And what she does that's quite a bit different is she actually creates silver nanoparticles, so extremely small particles of silver that she embeds in glass. And even the techniques she used to make the glass are from hundreds of years ago. And so if you have enough of these particles, they actually do the same thing as some of these thin films, and they actually give this iridescent coating. And so I'm sure it's harder to see unless you're right up here in person, but this is to think that this is actually microscopic particles of silver embedded in glass to create this color variable work is pretty awesome. I actually first saw her work when I was a postdoc out in Salt Lake City. There's a science and art museum there called the Leonardo, and she had one of her pieces through the looking glass there. So that's where I first saw some of her works.
Nancy said she's watching, I think, from Ecuador. She said many butterflies and moths here in Ecuador look brown and boring and then flash beautiful iridescence when they shine light.
Yeah, so that's one of the, so there are lots of hypotheses as to why animals might be iridescent. Obviously for lots of things like hummingbirds and different birds, the birds of paradise, it's for signaling. So they have these bright flashes of color to serve as a warning, to serve as an attraction signal. But there's also a paradox of iridescence in that if you are super, super bright and shiny, it might be really good for attracting mates. but it's also going to likely attract predators too. And so there's this cost behind iridescence to where you hope that you are much more appealing and intense just to the right things. And so you'll find that a lot of the reason we're looking at these different visual systems is to figure out who these signals are for. So it may be the case that a green beetle might be brilliant to other green beetles, but to another visual system like a bird, maybe it's not tuned appropriately and that green beetle will sort of look brownish. So we're going into the Soupçon Gallery, and this is a gallery where the LASM was hoping that we could showcase some of our specific science here at LSU. And so this room is largely collaboration from my graduate students and a couple other faculty at LSU and a couple other collaborators to showcase the beauty behind iridescence in the natural world of beetles and butterflies and shells and bird feathers, as well as how that relates to fashion, video games, and even LSU football. So let's take a look. So the first thing is an interactive chance for anyone coming to the exhibition to look at it in person. These are actual specimens that were used in the images here on the wall. And so one of the first steps is we have worksheets where kids can step up on the ladder and they can try to find the actual specimens and predict which ones go to which. And so the idea behind this room is to take you through in sort of a visual tour of how our specimens are used to teach various concepts of physics and art and science. So this first area here is called Captivating Iridescence, and the sole purpose of this wall is just to showcase how beautiful it can be if you're willing to overlook the fact that some are creepy crawlies. We've got bird specimens that were loaned from the LSU Museum of Natural Science. We have beetles that are from our collection or that have been loaned or imaged in various trips. And this is just to showcase the sheer array of colors, of patterns on these specimens. And so these beetles can be right here in our own very backyard. This is a beetle that one of my students, Abel Chow, collected here in Louisiana. Conversely, we have some beetles that are only found in Madagascar, like this purple one here, very LSU purple, all the way to places that are exotic like Borneo or in Malaysia, which is an example of this praying mantis here. So these are just images of either close-ups of the specimens or the entire specimen to demonstrate how incredible these can really look. But the first step in understanding why they have these colors is to take a look at the ultrastructure, or to zoom in quite a bit and get a sense for how these colors are made. And so this wall here discusses or showcases the different structures that are compiled to create this overall iridescent scene. So whether it is bird feathers that look a lot like Christmas trees, or concave dimples that act like colored mirrored bowls that spread light, or whether they're individual hairs or scales that look like convex mirrors, or even in this case, these are also hairs of a beetle, which is sort of weird, but these hairs are flat. And if you were to cut open one of those hairs, you'd find a crystal. And those crystal structures are actually what make that appear to be like little gemstones. So whether it's color gradients going from this dark purple all the way up to a lighter green to yellow, or stark contrasts of various barbules of bird feathers to the scales of a morpho butterfly, you can see that the creation of these colors comes in a variety of shapes and sizes. And so one of the first steps we do is to talk about and understand the structures themselves and how they might create those colors. So you can see here in the center of the room, this is a dress that was made by Dr. Casey Standard, who's in the College of Agriculture in the Textiles, Apparel, and Merchandising Department. And she made this dress here to be period specific. And so this dress is obviously with a little bit of our own take on it, is something that you might have seen in the Victorian era. And so when people, especially in high fashion, were moving from more rural areas to cities like London, they wanted to make sure they retained a connection with the environment. And one of the ways that they did that was adorning dresses and other garments with natural elements, whether it be peacock feathers or, in this case, jewel beetle wings. And of course, these are the original versions of things that we use quite often now, things like sequins and shiny fabric itself. So this is an awesome, awesome dress she made for the purposes of this exhibition to showcase how bio-inspired or biological materials can be incorporated into art and fashion.
Someone was joking saying it's a gold or blue, the whole dress.
Yeah, well, that's right. So that's an example of perception, and I use that meme quite a bit in some of my talks to showcase how it really does come down to the eye of the beholder and what environmental condition you're in. Yeah, and so over here we've got a work that was created by my graduate student, Sierra Weir, and she is an artist herself as well as a scientist in my lab. And whereas most of us work on structural color, which are the things that look metallic that change as you move, so example, this green puncture, the green part inside, that would be a structural color. She's interested in the other side, and that's the pigmentary colors. And so this bright red here is actually because of a pigment. And so what Sierra did is she wanted to show the two different types of data arising from the two different types of color classes we have. And so here is something that she created where the top is a data point on this specimen here showing the reflectance of light. And so this is a reflectance spectrum curve. And that's when light bounces off this metallic looking structure. But on the other side, she has created an absorbance spectrum. And so if you were to measure the red portion of this same beetle, rather than light bouncing off, the vast majority of light gets absorbed. And so this is the absorbance spectrum, and she made this and dyed this material from actual insects. And so that's how the original red dyes were made, and a lot of products to this day are still derived from insects. And so she wanted to make this work as natural as possible. So here she is sort of discussing the difference between pigmentary and structural color and allowing her own artistic abilities to shine through as well. So this wall is the science of the ultrastructure. And so my graduate student, Abel Chow, is trying to figure out how these actual structures produce these colors. And so what these are close-ups at extreme magnification, anywhere from 100x to all the way up to 50,000x or more, 100,000 times life size. And these are the actual structures that comprise these beautiful colors. And it's sort of odd, but in a lot of ways, these structures themselves don't have a color. They're clear, or they're just like your fingernails and sort of translucent. But it's not what the structure is that creates the color. It's how light bounces off. And so what you get is, this is an extreme close-up of morpho butterfly scales, those brilliant blue scales. And if you zoom in even farther, you get down to this, and you can see that a single scale that you can't even see with the naked eye has a series of these cross ridges and these angles, and all of these serve as a physical structure. So if you think about how when you were a kid and you may have had a prism and you got light to pass through a prism and then display a rainbow on the other side, even though the prism is clear, you got to see all the colors of the rainbow. And this does a similar function where all of these photons of light are striking and bouncing off these ridges and sometimes striking each other on the reflection, and that is what is giving us this color shift iridescent signal. So yeah, it's interesting to see that although these were actually not formed, these images weren't formed from photons of light, they were formed from electrons passing through or bouncing off the structures. It's actually semi-transparent relatively clear scales, feathers, in this case, multi-layer stacks that cause these incredible colors. So then here we just have an overview of how you go from a beautiful, crazy, iridescent beetle that you wonder why in the world would it have a blue body and a red butt, to how we take this and transform it into our studies of scientific data. And so this is an example of a jewel beetle called Democroa gratiosa, and as you can see, if you can imagine closing the wings and just looking at the top of the beetle, so you wouldn't see any of this blue, it sort of looks like a typical bug. It may be a little shiny and green and has this nice white non-structural color patch, but for the most part it's just a green bug. But when it opens its wings, it displays this brilliant blue backside, and most beetles don't have this. You open up the wings of most beetles and these are more or less just sort of brown and clear. But these beetles are clearly using this as some form of signal. Maybe they sit on a leaf and open up the wings and flash this like a signal, or maybe it's for when they're in flight. You flip the beetle over, and then you get to really, really see the showcase of iridescence and color that it has.
We had a question for you. How do you get such amazingly clear resolution at such high magnification?
Yeah, so in my case, photography is one of our data sources. And so here at LSU in the Department of Entomology, I set out to build an incredible imaging studio that is capable of high-resolution images from the organisms themselves. They can take pictures of whole drawers of insects all the way down to the macro scale. And so we have a tremendous partnership with a few people that are sort of the innovators in the field of digital photography and macro photography, and we've created an imaging studio, and we in my lab spend lots of time trying to keep our skills honed and to really be able to get these images given the instrumentation we have available. So yeah, so you can take these two beetles, and if you zoom in on just this little blue part, this is what you see. And this is sort of this wrinkled, in science we call it rugose, this wrinkled rugose surface of these brilliant blues. But if you focus on another area here, like at the tip of the wing, or the hardened wing called the elytra, you can see that you get these big ridges and these series of dimples, these impressed punctures that serve to scatter light as well. You then go down into the SEM to look at these punctures, and then you realize that there's a single hair coming out of each of these. And so this is just a magnification of about 500 times life size. But what we also do is we want to understand how these colors are made, and so we'll actually cross-section these and use a device called a TEM, or a transmission electron microscope. And what you see here is this sort of boring looking stacks of layers. But as my students can tell you, and this is the work of Abel Chow, one of my graduate students, these layers, the numbers of layers and how thick they are, actually give the color that they do. And so again, light passes through various depths and bounces out, and you're able to create or predict or model different colors based on that. So this is showing the red portion and the green portion and the blue portion of these beetles, and so we can actually figure out the math behind the color in these natural specimens. So then we've got something that's really fun. This is another one of the ways that this whole exhibition first started, and this was my first year at LSU, so that was 2018, and I went to the LSU Mississippi State game, and I was sitting in the audience, and LSU came out in these atypical helmets. Now, for any of you that are Tigers fans, you know that this is not something that they wear every day. In fact, they've only worn these helmets a grand total of one time. And they did this to commemorate the 1918 season that was not played due to World War I. And so what LSU did is they wanted this helmet to be reflective of the culture that is Louisiana, and they tried their best to create a paint that would resemble a Mardi Gras bead. And of course they wanted it to change from LSU purple to LSU gold. And so they actually worked with Nike, and they also worked with a special paint shop that does motorcycles in California to try to create this iridescent paint. And so rumor has it the paint cost about $1,500 a gallon for something they only wore once. And I saw that, though, and I thought this is a perfect teaching tool given what we do. It's armor, just like beetles. It's like an exoskeleton. And it also, in addition to football, which I very much enjoy here at LSU, it's an actual representation of what we're studying in the form of iridescence. So yeah, so I had one of my boss's bosses was able to talk to people in athletics and get one of these helmets on loan so that I could showcase this in the exhibit. So yeah. I hope that you guys, for those of you tuned in, have enjoyed this behind-the-scenes tour of iridescence. We hope you come see it in person. It will be here until next July.
Where it's at again.
Sure. So this is at the Louisiana Art Science Museum, which is downtown Baton Rouge on River Road, and right across from the old State Capitol building in the old train depot. We're going to try to have several other events throughout the year. We're going to see if we can't get some of these other featured artists like Chris Marley or Francesca Schenk or other people to come out and actually discuss their artwork in sort of special, more intimate situations where they can take us through the true meaning and the science behind their art. So yeah, it's going to be running through July 2022, and we hope you come out and see it.
Thank you. And can you tell everybody that it's going to be recorded and put up on the LSU AgCenter YouTube?
Sure, yeah. So this is also being recorded and will be put up on the LSU AgCenter channel YouTube.